Assalamualaikum and hi everyone. For this assignment, we are going to explore next generation sequence in thoracic aortic aneurysms and dissection disease. Before that, I would like to introduce our team members and myself. My name is Ni Muhammad Irfan HAMIZAN bin SUPIAN, and my team members are BATRISYIA ZAIDI binti BAHARUDDIN, EZZATI binti ARBAIN, Muhammad Haziq Qayyum bin ZAIDI, and Siti NASHRAH binti SUBKI. In this introduction, we will explore how next generation sequencing is transforming our understanding and management of thoracic aortic aneurysms and dissections, highlighting its impact on early diagnosis, risk stratification, and the development of target therapies. Actually, what is thoracic aortic aneurysms and dissection, or we call as TAAD? TAAD are life-threatening conditions that involve the weakening and tearing of the aortic wall within the chest cavity, as stated by Qayyum et al. 2021. These conditions can lead to catastrophic outcomes if not diagnosed and managed promptly. Historically, the understanding of TAAD has been limited by the complexities of its underlying causes, which often involve a combination of genetic and environment factors. What is next generation sequencing, or we call as NGS? NGS is a powerful technology that allows for the rapid and comprehensive analysis of genetic material. Unlike traditional sequencing methods, NGS can process millions of DNA fragments simultaneously, providing detailed insights into the genetic underpinnings of disease, as stated Genetic Analysis Education Journal. This is crucial part. What is the relationship between TAAD and NGS? In the context of TAAD, NGS has enabled researchers and clinicians to identify genetic mutations that predispose individuals to aortic aneurysms and dissections, enhancing our ability to diagnose, monitor, and treat these conditions, as stated by Higgs et al. 2018. By leveraging NGS, we can now screen for a multitude of genes associated with TAAD, uncovering both known and novel mutations. This genetic information is crucial for early diagnosis, family screening, and personalized treatment strategies, ultimately improving patient outcomes. Additionally, NGS-driven research is uncovering new molecular pathways involved in TAAD, paving the way for innovative therapeutic approach.
Sample preparation is the process of getting DNA ready for next generation sequencing NGS. This requires a few methods. First is nucleic acid extraction, the process of purifying genetic material from an initial tissue sample as a starting point for NGS library preparation. DNA extraction involves isolating cells from tissue and then breaking or solubilizing the cell's plasma and nuclear membrane to release the genetic material. A variety of methods are available, including phenol-based extraction, chloroform-based extraction, spin column magnetic beads, and single-cell isolation. Fresh starting material is always recommended, but this is often not possible, so samples should be stored appropriately, which usually involve freezing or cooling at specific temperature. Second is library preparation. A sequencing library is essentially a pool of DNA fragments with adapters attached. DNA library preparation for NGS involve fragmentation, which involve breaking DNA strand into pieces using physical, chemical, or enzymatic methods. Adapters are chemically synthesized oligonucleotide attached to a DNA molecule to act as barcode. The optimal ratio is 10 adapters to 1 fragment, as too many can cause adapter demers and lower sequencing efficiency. Library quantification is crucial for obtaining high-quality NGS data and can be done using techniques like UV adsorption, intercalating dyes, hydrolysis probe, and droplet digital emulsion (PCR). Properly removing adapter dimers and fragments of the wrong size can improve sequencing efficiency and quality.
The third step is sequencing and imaging. The amplified DNA fragments are loaded onto a sequencing platform. The library fragment acts as template of which a new single-stranded DNA fragment is synthesized. The sequencing occurs through a cycle of washing and flooding the fragment with the known nucleotides in a sequential order. As the nucleotides incorporate into the growing DNA strand, they are digitally recorded as sequence. The MISAC detect fluorescence caused by the integration of fluorescent-labeled nucleotides into the developing strand of DNA. Once sequencing is completed, the raw data must undergo several analysis steps. A generalized data analysis pipeline for NGS data includes pre-processing the data to exclude the adapter, sequence, and low-quality reads, mapping the data to a reference genome or de novo alignment of the sequence read, and analyzing the compiled sequence are all steps in generalized data analysis workflow for NGS data. The sequence reads are aligned to a reference genome to identify genetic variants. Identification of somatic and germline mutation event that may aid in diagnosis of a disease or genetic condition is one of the aspects of analysis. Specific genes associated with TAAD such as FPN1, COL3A1, TGFBR1, TGFBR2, SMAD3, and ACTA2 are closely examined for pathogenic variants.
Findings and implication of using NGS. First, genetic etiology identification. Recently, NGS for the molecular diagnosis of TAAD has emerged as a useful screening technique to find gene mutation linked to the disease, giving the patient and doctor a chance to step in and stop emergencies for patient and their family. This shows that the complete identification of genetic alteration linked to TAAD is made possible by NGS. It has the ability to identify changes in several genes at once, many of which are connected to familial TAAD and connective tissue illness. For example, TAAD has been linked to the mutation in the genes FBN1, COL3A1, ACTA2, MYH11, and SMAD3. This aids in comprehending the underlying biological process and inherited patterns of the illness. 2. Early diagnosis and screening. Early diagnosis is made easier by the use of NGS, particularly in those who had a family history of TAAD. Early detection of harmful variation allows for closer individual monitoring, lifestyle adjustment, and early intervention to avoid catastrophic outcomes like aortic dissection. 3. Personalized medicine. By adjusting treatment plans and surveillance tactics in accordance with each patient's unique genetic profile, NGS help to advance personalized medicine. For instance, compared to individuals without such mutation, people with certain genetic variants could need more regular imaging tests or preventive surgical procedures at lower aortic diameter. 4. Stratification. Clinical professionals can more accurately stratify patient likelihood of acquiring TAAD by recognizing certain genetic variants to prevent complications. More intensive surveillance and early surgical intervention may be necessary if certain mutations are linked to a more aggressive cause of the illness. Improving family counseling. The genetic counseling provided to TAAD affected families is enhanced by NGS results. It is possible to screen relatives for known harmful mutations, which determine their risk and help choose the best course of action for therapy and surveillance. In families whose inheritance patterns are autosomal dominant, this is very crucial. 6. Discovery of new genetic variants. NGS keep finding new genetic variation linked to TAAD, increasing the number of genes linked to the illness. This continuous process of discovery contributes to our understanding of the genetic basis of the disease and may provide new targets and approach for treatment.
So right now, I will address the advance and pitfall of using next-generation sequencing NGS to detect thoracic, aortic, aneurysm, and dissection TAAD. It is crucial for us to recognize and address its limitation in the context of TAAD detection to avoid any mistake to occur. Limitation 1. Unable to identify every kind of genetic variation. Hartman P. et al. In 2019 state that current short-read-based NGS approaches have significant diagnostic limitations for detecting deletion and duplication in focused NGS data. This might lead to misdiagnosis in TAAD patients. Limitation 2. Production of numerous variations of unknown significance (VUS). From the research from Papadopoulou et al. In 2023, another critical constraint of NGS analysis is the generation of a large number variant of unknown significance (VUS), complicating the interpretation of genetic data and clinical decision-making in TAAD cases. Limitation 3. High degree of technological proficiency is necessary to conduct NGS. Papadopoulou et al. In 2023 state that the application of NGS for clinical purpose demand a high level of technological expertise and experience, posing a challenge for widespread implementation and accurate genetic analysis in TAAD cases.
Now, I will continue with the solution for its limitation that has been mentioned before. Solution 1. By implementing highly sensitive NGS technologies, we must adopt highly sensitive NGS technology capable of detecting all genetic variation, including intragenic copy number variation. By leveraging advanced NGS technology, we can enhance the accuracy and comprehensiveness of genetic testing for TAAD. Solution number 2, we need to establish variant registry and data exchange. It is imperative to establish this based on the evolving knowledge. By exchanging data across laboratory and leveraging collective expertise, we can enhance the accuracy of the variant interpretation and minimize the impact of VUS in TAAD diagnosis. Solution 3, we need to ensure technological proficiency and validation. To ensure the reliability and sensitivity of the NGS genomic analysis, laboratory must maintain high level of technological expertise and validate their methodology frequently. By upholding standard of excellence and expertise, we can enhance the quality and reliability for genetic testing for TAA.
In conclusion, next generation sequencing, or we call as NGS, has revolutionized the field of thoracic aortic aneurysms and dissections, TAAD, fundamentally changing how we approach these life-threatening conditions. By providing a deeper understanding of the genetic factors involved, NGS is enabling precision medicine approach that improve diagnosis, treatment, and ultimately patient outcomes. As technology and research advance, the role of NGS in combating TAAD will only grow, bringing new hope and improved health to those affected by these serious conditions. I think that's all from us. Thank you.